I'd like to start by welcoming you all today. This is a picture of my daughter, Chloe. Sorry, I've just lost something.
Kath, there's that take control button. If you just click that one, you should be able to see what you're looking for.
Okay. All right. So, look, our whole team is delighted to be here for another exciting Prader-Willi research update. And this is the 5th year of these webinars for us. I want to welcome everybody tonight. You're all part of our Prader-Willi community, whether you're a person with Prader-Willi syndrome, a family member, a researcher, a clinician, a professional carer. We're all here as part of this one community tonight. And though we're spread across this continent and even around the world, although mostly our get-togethers are digital, we're still very much part of this one community. And it's together that we understand and care about the unique challenges that Prader-Willi syndrome presents and how we can work together to tackle these challenges head on and ultimately transform life for our beautiful people with Prader-Willi syndrome. And this beautiful 3-year-old girl smiling at you tonight is dressed up for her sleep study. And if she reminds you of me in any way, it's because she's my daughter. And when Chloe was diagnosed with Prader-Willi syndrome at 19 days, it was the first time I'd ever heard of this condition. However, she's the catalyst and the inspiration for the establishment of this foundation. The work, though, is not for her alone. Prader-Willi Research Foundation's work is for every member of our community, for all of our families and all of the beautiful people who live with Prader-Willi syndrome. And every day our work is dedicated to you. We'll continue to work hard with relentless focus to drive the development of treatments and bring them to our community here in Australia. There are many people in our team that make this vision become a reality, and I want to pay tribute to all of them tonight. Firstly, to everybody who donates, volunteers or raises awareness, thank you. You enable this work to happen. In our team, which includes Dr. Diane Webster, our research director, Debbie, our family liaison, Nicole, our ops coordinator. Thank you all for every day, the amount of work you put into making our goal come to life. And I also want to thank the clinicians and researchers who put in the hard yards, that advocate for more work and more services for our kids and adults. And some of them take significant career risks because they want to make a difference. And for many of you don't know that we also have a board of directors who are professionals from across Australia. Some have direct Prader-Willi relationships and some don't. And these people have put in a lot of work every month, some of them for 8 years or more, from the goodness of their hearts, and I want to acknowledge and thank them also. Together, we've been able to build real momentum in Prader-Willi research in Australia. We're really proud of the momentum that has built here. To think just 8 years ago, our organisation was only just getting started and there was less than $300,000 of Australian investment into Prader-Willi research. We started from scratch and it has been families who funded us and enabled us to do that. The first Prader-Willi conference, the first PWRFA conference, research conference, was actually bankrolled from my mortgage, and then a family from Gippsland auctioned a cow and then kept re-auctioning that cow until it earned $15,000 for us to kickstart some projects. And today we have some globally significant projects right here in Australia, including projects that other rare conditions just dream about. We have got here through carefully selecting projects, developing a minimum data set, and then supporting those projects to go on and attract more funds. And in doing this, we've developed our own indicator of success, which is a 10 times multiplier of funds invested to new funds attracted. So for every dollar we've invested in research projects across our whole portfolio, on average, that dollar attracts another $10 investment from another funder. And we think that we're very proud of that. We think it shows that we know how to pick the right projects that can follow a pathway to get to treatments for Prader-Willi. We also understand the importance of clinical trials to families, and especially as a pathway to accessing treatments. And so tonight we have two fantastic speakers to talk you through new clinical trials available in Australia. Firstly, Associate Professor Tania Markovic and her team, who have been awarded the PWRFA's 2023 grant to investigate tirzepatide in Australia. Tirzepatide has received significant interest internationally for its diabetes and weight impacts. And then Dr. Diane Webster, our Research Director, will update you on the latest clinical trials, including, as I mentioned before, an exciting new clinical trial announcement. So with that, I'm going to hand you over to Associate Professor Tania Markovic to tell you about the project she's leading on tirzepatide.
Thank you very much for that introduction, Kathleen. And of course, we are very grateful to have received the funding for this study, which we think is very important. And so what I'd like to try to do briefly, Kathleen, tell me if I've gone over time, please, is to tell you a little bit about the study, why we're doing it, and then hopefully have some time for you to ask questions. So the title of the study, you can see it's written there, Gastrointestinal Transit and Safety of Tirzepatide in People with Prader-Willi Syndrome. Here's the title again, and this slide just really shows our co-investigators, and I'm very proud of our team. We're very fortunate to have Karen Jones and Michael Horowitz involved. They are world experts in gastrointestinal incretins and how they affect the gastrointestinal tract, and which is very important for our study and for people with Prader-Willi syndrome. Whoops, let me just get this right. Yeah. So why we took, oh, what have I done? Did I do something wrong? My slides have disappeared.
No, that's okay. We'll get them back.
Did I press the wrong button? Anyway, while you're getting them back, I can talk. So I'm talking about am I pressing the wrong button maybe?
If you just click the screen, it should
okay,
trickle through. There you go. You can take control again, Tania.
So I've been clicking this arrow down the bottom.
It's ready for you to take control when you're ready.
I have to take control, sorry. Take control and just click the screen. No, should have practiced this. Sorry, let's go ahead. Oh. Now this is a bit unprofessional of me. Okay, here we go. So, so first of all, I just wanted to discuss incretins. Now, the reason is because these drugs, tirzepatide, is a synthetic version of a hormone, a type of hormone that we make that are called incretins. These incretins are hormones that we all make in our gastrointestinal tract. And they are secreted rapidly after a meal. They have an important role in signaling to the body and the brain about nutrients entering the gastrointestinal tract. One of the first roles that they were found to have many years ago now is that they regulate pancreatic insulin release in response to elevated glucose levels. And this is what we call the incretin effect. They also have effects on the motility of the gut, on the absorption of nutrients, and on food intake. There are two main incretins that have been found. One is called glucose-dependent, got a long name, insulinotropic polypeptide, which we shorten to GIP, and this is secreted by cells in the lining of the upper gut. And then the second one is called glucagon-like peptide 1, or shortened to GLP-1, which is secreted by cells in the lower gut, in the lower small intestine and colon. So these drugs have been around, synthetic versions of these drugs have been around for a long time and have been used in diabetes. And what we know is that in type 2 diabetes, the secretion of GIP-1 and GLP-1 after a meal is normal, but the pancreas does not respond to the GIP and GLP-1 as well as in people without diabetes. So there is lesser of an insulin release. And when you give a synthetic version of GLP-1 in a pharmacological dose, there is if the glucose levels are high, above normal, there is a stimulus to increase insulin release. So a very useful drug for diabetes because you're getting you're getting glucose levels dropping when they should be dropping. You're not getting low glucose levels. And an additional benefit that was found with using these drugs is that there was weight loss. Not a huge amount of weight loss, but more weight loss than with any other drugs used to treat diabetes to date. So with the earlier agents, in the order of 3 to 4% body weight loss. And the early agents, some of you may have heard of, exenatide was the first one. It was a twice daily. These are peptide hormones, proteins that generally need to be given as an injection because they would be digested by the gut. There is actually an oral version of one of these drugs, but much higher doses are needed for it to be effective. So in general, best given as an injection. So the earliest one was exenatide, which was the trade name was Byetta. Then there was a long-acting version of exenatide that could be given once a week called Bydureon, not available anymore. The next one that came out was called dulaglutide or Trulicity, and it's still available. Then after seeing that these drugs were good for weight, further studies were done by pharmaceutical companies, and they found that if you could push the dose even more, which you can with certain compounds, you can get quite substantial weight loss. So the first type of incretin, which was which was a GLP, these are all GLP-1 copies or analogues that I'm talking about, was liraglutide, which is a once daily injection. It's the first one that was marketed trialed in people with obesity and marketed for obesity. And it is used in a low dose for diabetes because a lower dose results in good glucose control, and pushing the dose higher didn't have a major effect on glucose levels, but a higher dose did result in good weight loss of around 9% body weight loss. And that's called Saxenda, and that's available here and approved for obesity management. Semaglutide is the one that a lot of you, well, everybody would have heard about because you can't not hear about it, TV programs and everything about this drug, because the weight loss with this drug is even larger, on average 15% body weight loss in people with obesity, a bit less in people with diabetes. People with type 2 diabetes always seem to get less weight loss with pharmacological treatment. There are two versions, well, not two versions, two products. One is called Ozempic, and that is the lower dose of this drug, which is used for diabetes and is available here. The higher dose, which is called Wegovy, is 2.5 times the diabetic dose, and that is the dose that was used in the weight loss trials. It's been approved for use in Australia, but isn't available here yet. I'm told it probably will be at the end of the year, and that's because these drugs have been so popular. There's been a shortage of them. Now, tirzepatide, the compound that we want to study, is a combination of GLP-1 and the other main incretin, GIP, that are combined together into a single molecule. So it's called a dual agonist containing these two incretin hormones. And it results in even better improvements in glucose levels in people with diabetes, and even greater weight loss in the order of, in the clinical trials, 21% body weight loss, which is approaching what we see with bariatric surgery, weight loss surgery. So quite remarkable. This drug, the starting dose is 0.25 milligrams once a week, and then the dose, it's gradually, all these drugs are started at a low dose and gradually increased to minimize side effects, which I'll talk about. The dose used in the weight loss trials was 15 milligrams per week. Currently in Australia, only this medication has only been approved for use in people with type 2 diabetes, is available here. Supplies have again been problematic. It's called Mounjaro, the diabetes product. Exactly the same drug, same doses. So unlike the previous, the GLP-1 analogues I mentioned, this one comes just in the same doses are used for diabetes and weight loss, because you get more bang for your buck as you increase the dose, even in diabetes. And the weight loss product is called Zepbound. It's been approved for use in the United States, but has not been approved by the TGA here yet. Now, these drugs, as I've mentioned, a lot of dramatic effects on weight, which initially we thought was due to their effect on the gut, but we now know that there are GLP-1 and GIP receptors in the brain, and importantly in the hypothalamus, which is a very important part of the brain for control of appetite. And these funny little squiggly things are meant to represent the receptors on which these hormones bind, and when they bind to these receptors, they stimulate hormones and signals in the brain that reduce appetite, and they inhibit. So these are the ones that stimulate appetite, and they inhibit other receptors. So they stimulate pro-opiomelanocortin, which reduces appetite, and they inhibit neuropeptide Y, which results in a reduction in appetite. So, and when we first started using these, we didn't know exactly how they work. We still don't know exactly how they work, but they do have effects on the brain, which in areas that control appetite. So it all makes sense that this fits in with their role on reducing appetite. And also, I should have mentioned, they affect parts of the brain involved in craving, the enjoyment from the craving for food, hedonic, which sort of means the enjoyment for food, and there is some evidence that they reduce people's cravings as well as their desire for alcohol. So interesting, very interesting effects. Now, there are not only these GLP-1, GIP receptors in the brain and the gut, of course, and the pancreas, but throughout the body. And there are studies have been done exploring some of these effects, and it's been shown that there are ongoing trials with tirzepatide, but certainly with the dual compound, but with GLP-1, we found that there are improvements in kidney function in people with diabetes, reduction in cardiovascular disease in people with diabetes, and just towards the end of last year, significant reductions, 25% reductions in cardiovascular events in people with obesity, and a history of cardiovascular disease. There's also been some very early studies suggesting they might result in improvements in Parkinson's disease, and there are trials for both the GLP-1 and the combined compound looking at effects on Alzheimer's disease, and so it's very interesting. A very interesting class of drugs. But let's see if I can get through. But what we are of course very interested in what effect we're all very interested in what effects these drugs will have on appetite and body weight in people with Prader-Willi syndrome. But we are concerned and from a safety perspective want to look closely at the gastrointestinal tract, because these hormones are made in the gastrointestinal tract and have important effects on the gastrointestinal tract that are really to help us absorb nutrients, but in pharmacological doses can be problematic. We know that the GLP-1 slows gastric emptying, and it's been postulated that might partly contribute to the glucose lowering effect. And you can imagine that slowing gastric emptying allows nutrients to be absorbed better, mixed better, and but if you overslow it, that can cause symptoms, and it's undoubtedly partly related to some of the gastrointestinal side effects that people experience. The typical side effects are nausea, diarrhea, and constipation, and some people can get both diarrhea and constipation. And it seems that if you build the dose of these, well, not seems, we know that if you increase the dose very gradually, and generally the dose is increased monthly, that reduces the chance of side effects. Now, what have I said? With the longer acting agents, you do get what we call a tachyphylaxis, where you no longer experience these side effects. The body sort of gets used to it after a few months when you gradually increase the dose. The GLP-1 analogues also, I just wanted to, this picture of the gut here is, so they have effects on the stomach, they have effects on the duodenum and the upper small bowel, where they reduce intestinal motility, and by doing that they reduce the absorption of nutrients. So you end up having more nutrients going down, sorry, that's the small bowel there, more nutrients that are undigested and unabsorbed going into the large bowel, and that may contribute, this is in the pharmacological high doses, and that may contribute, it probably contributes to the diarrhea, because you're getting unabsorbed nutrients in the large bowel. But also there is reduced activity in the bowel, throughout the bowel, that probably contributes to constipation. One interesting finding is that there are tumours. People can have tumours where they overproduce GLP-1, and these tumours are associated with a marked delay in colonic transit, in food going through the colon, and severe constipation. So we know some things about these GLP-1s, but there's a lot we don't know. And in fact, if you look at the actual data, there's very little data on the effects of incretins on bowel motility, even in people who have neither diabetes nor certainly none in people with Prader-Willi syndrome. So we don't know much. We don't have hard data on how these agents affect the gut, let alone people with more with other conditions. Oops, keep pressing the wrong button. So what do we know about how these drugs work in people with Prader-Willi syndrome? Can we expect that that something like tirzepatide will result in the sort of weight loss that we see in in people without in the general population. So I just want to, these are people not familiar with these graphs might find them a bit difficult to read, but I'll just quickly walk you through it. So this is the result from weight loss in adults with obesity, just ordinary everyday obesity treated with liraglutide, that when they were on a placebo, so not on the active drug, people always lose weight always do well in a study. It's known that there's a placebo effect that actually works better than not being on any treatment at all. So they lost about 3.5% body weight if they were actually taking the drug, taking the placebo, whereas if they stopped the placebo, they didn't lose as much weight. And in those taking the active drug, liraglutide, there was a 9% body weight loss over 1 year. Now, the reason I showed this is because this is the only sorry, trying to navigate this, the only drug that has been studied well with a double-blind placebo-controlled trial in people with Prader-Willi syndrome, and the trial was done in Australia as well. Very small numbers of subjects, but in adolescents and children with Prader-Willi syndrome, and essentially what they found was that whether the children were on liraglutide or placebo, there was no effect, no difference, no significant weight loss in either group. So that looks like there might be weight loss, but it's only just over half a body mass index measurement, half that it's dropped, which is insignificant and no different. But they did find, interestingly, that the hyperphagia, the hyperphagia scores and the drive to eat were lower in adolescents who were treated with liraglutide. Importantly, the treatment was well tolerated and they had all the usual side effects that we see, but there were no worrying adverse events. But unfortunately, no significant weight loss. Now, why all the buzz about tirzepatide? I mentioned this at the beginning, and again, it's one of these scientific-looking graphs, but just this one's probably an easy one to look at. In this study, which went for a year in people with obesity, people who were on placebo lost 3% of their body weight. People on the 5 milligram, relatively low dose of the tirzepatide lost 15% of their body weight, so similar to the maximum semaglutide dose or Ozempic dose. When the dose was increased, there was greater weight loss, such that with 15 milligrams, there was 21% body weight loss with this drug. So clearly, yes, we're very interested in this. I don't know who's doing the little reactions, but they're sweet. So will this drug work in our patients with Prader-Willi syndrome? We know that hyperphagia, the drive to eat, in all of us is high, but in people with Prader-Willi syndrome, it's off the scales. And there's very different physiology going on in people with Prader-Willi syndrome. There's disturbances of the neurocircuitry in the hypothalamus, in the prefrontal cortex, in our brain, in the orbitofrontal cortex, so seeing food, how they react to food is quite different to the regular situation. They have, both when fasting and in the fed state, they have abnormalities in their reward circuitry, and their reward circuitry. And I don't need to tell you, you know more about it than me, I'm sure. So we're not dealing with, so when we're using a weight loss drug, we're not, we've got a much greater drive to eating. So clearly whether these drugs are going to work in people with Prader-Willi syndrome is a big question mark. Also, the other important aspect is that we know, just as we know very little about how these incretins work in the general population, how they affect the bowel, people with Prader-Willi syndrome you would all know, do have bowel problems. And we have, and it really is an area that hasn't been well studied. I've tried to look it up for a few patients of mine who've just suddenly, I've had a whole lot of patients who've had severe constipation, and I've been trying to review the literature to see, you know, how's this best managed, and it's that there's just very little known. So on this slide, I've summarized all the literature that I'm aware of in people with Prader-Willi syndrome. We do know that there can be a rare, thank goodness, but very serious, there have been reported very serious cases of massive stomach distension that can lead to stomach rupture and can be fatal. And these typically follow a very large binge of food. And I've had a patient, or at least two patients, who've had this. So we know, and there have been, these are quite old studies that have, not that old, sorry, 10, 15 years old studies in really just 20 patients on published data looking at gastric emptying in people with Prader-Willi syndrome. One study used a method that's not the optimal method to measure gastric emptying. It's called the paracetamol absorption technique, and it found that in these 12 people it was normal. A later study that was done in just 8 people using a nuclear medicine method, where people have a meal that's labeled with a tracer that can then be photographs are taken, and then you can follow the food going through the gastrointestinal tract. With this study, and this is the type of technique we want to use in our study, it's the gold standard, and it's one that Michael Horowitz and Karen Jones know a great deal more about than me. This was delayed in 5 out of 8 of the subjects. In the remaining 3 subjects, it was at the upper limit of normal. In a study that was done on some of our patients at the Garvan Institute, that unfortunately is just in abstract version, they studied 13 people with this same method, the scintigraphy, and they found a lower rate of just 23% with delayed gastric emptying. They then treated these people with exenatide, one of the early GLP-1 analogues, and they found that in the 10 remaining people, 3 developed significant delays in gastric emptying. So not as bad as one might have guessed. When we look at the lower bowel, there's even less data. Just one study that I found in 20 people and with Prader-Willi syndrome, 8 reported a history of constipation. 13 out of the 20, so 65%, were found to have fecal substance in their rectum, compared to, in a control group, just 3 out of 20, or 12%. And they looked at something called the gastrointestinal transit time, which was determined with radio markers in the gut. The time of greater than 3 days is considered to be significantly elevated, and this was in 24% of the patients compared with Prader-Willi, compared to the general population, where it's only in 2 to 4%. And I know that I've got a number of my patients who tend to pick their anal area, and undoubtedly, we used to think it's a very odd thing to do, but it is almost certainly related to constipation and having discomfort down there. And it can be really, it's obviously not a very nice thing to do, but it can be very disturbing and problematic for our patients and carers. What questions did we have as a group around incretins in people with Prader-Willi syndrome? The questions that we had were, will these potent new incretins promote weight loss in people with Prader-Willi syndrome? Will they be safe? And even if they don't promote the sort of weight loss that we're seeing, or not much weight loss, will they actually be helpful for management of type 2 diabetes in people with Prader-Willi? So let me just... which leads us to our study. So these are the important questions that we are hoping that we will at least get some answers, some definitive and some will be preliminary, which leads to to our study, gastrointestinal transit and safety of tirzepatide in people with Prader-Willi. So our aims are to assess gastric emptying. In addition, which hasn't been done in any studies, cecal arrival time, which is a measure of small intestinal transit, so the movement of food through the upper bowel, the small intestine, and also colonic transit time, movement of food down to the rectum in people with Prader-Willi syndrome. We want to assess this both, we want to assess how tirzepatide affects gastrointestinal transit, gastric emptying, all these measurements we want to do both before and then after the patients have been on tirzepatide, to see whether it causes significant problems as far as gastrointestinal health goes. And then, so we think that with the study we've designed, we'll be able to answer those questions. We will also get an inkling of whether Prader-Willi might have a beneficial effect, whether tirzepatide might have a beneficial effect on body weight, on food-seeking behaviours and metabolic markers in people with Prader-Willi syndrome. And very quickly, I've just about finished, just to show you how we're going to do this. It's a short study, 3-month study. This is because that's all we think we need as a minimum. We don't want to waste any of the money that we're given, that you've very kindly given to us and raised for us. So we've tried to design a study that can answer the questions with the least amount of expenditure. So a 12-week trial, we're going to increase the dose. We're going to start the dose. We're going to do these sophisticated nuclear medicine scintigraphy trials before starting tirzepatide. We're going to measure gastric emptying, small intestinal and colonic transit time. We're going to do stool charts, and there's a constipation questionnaire that we're going to do on our patients. They'll be doing at home. We will obviously be doing a physical examination, and before starting the tirzepatide, we'll be doing Prader-Willi specific behaviour and hyperphagia questionnaires, as well as fasting blood tests to look at glucose, cholesterol, lipid levels, and metabolic parameters. Then patients will start tirzepatide with a 2.5 milligram dose. A second study will be done after just 1 month on the low dose, the gastric emptying study. This is primarily a safety study to make sure that we're not causing significant delays in gastric emptying, and if we are, then depending on the degree, we'll decide whether or not to continue. That patient will continue, whether they're having symptoms, side effects, whether we'll do X-rays to check if there is gastric distension. So, quite rigorous safety measures to determine if it's safe for a particular individual to continue. The stool charts will be repeated, adverse event screening and physical exam at 1 month. If all's going well, the dose is increased, and the patients then come back after a month for examination at our centre, physical exam, adverse event screening, the constipation questionnaires, etc. And then at the end, at 12 weeks, at the 2-month mark, the dose is increased to 10 milligrams, which is a pretty decent dose of tirzepatide, and we think adequate to look at efficacy and safety. And because you might recall that going from the 5 milligram to the 15 milligram dose, there's not a huge difference in weight loss. There is an extra 6% weight loss in people without in the trials, and people without in people with obesity. So we think that'll give us a good idea about a good hint about efficacy. And then importantly, we're going to repeat all those the gastric emptying study again, and the small intestinal and colonic transit time, to see the effect of the drug over time on gastrointestinal function, and then examine the patients, etc., as before. And that is, and then I've got, I think, my last slide. So these are the potential study outcomes where we're hoping that this trial will teach us more about bowel function in people with Prader-Willi syndrome, that will help guide treatment for problems such as constipation, gastric dilatation. This trial should provide us with data on the safety of tirzepatide in people with Prader-Willi syndrome, at least preliminary data on the safety, as well as preliminary data on the effects of tirzepatide on appetite, behaviour, weight, and metabolic parameters. And if the results look promising, then it's this type of pilot data that is very strong and makes an application for an NHMRC, one of our government National Health and Medical Research Council grants that will provide funding for a larger trial where we can really look at larger numbers of people to have a more definitive study looking at safety and efficacy of the drug in people with Prader-Willi syndrome. If we try to apply, which is why we're so grateful for the money from this organisation, if we had tried now to apply for funding, we'd be knocked back very quickly because it's very, very hard to get this funding. But if you've got good pilot data, there is a schema for rare diseases, and we think that we stand a very good chance with pilot data. So let's hope we get there. Thank you very much. And I've gone way over time, I'm sorry.
That's fine. Tania, thank you so much for sharing all that with us tonight. This is such an important area, and we are absolutely delighted to be working with you and to be really specifically addressing this question. So globally, people are monitoring tirzepatide, but this is the only study that I'm aware of which is actually setting out to really specifically and deliberately look at those safety aspects, which is so important. So thank you. If anyone's got questions, pop them in the Q&A. We'll probably only have time for one or two, but it's always good to see what the questions are and we can address them in other forums. Tania, one of the questions that came up as we were discussing the study, so this is a question from a family member. They're interested to know whether you're going to be doing any carer-based questionnaires as part of the
Yes,
evaluation measures.
Yeah, good question. Look, those behavioural questionnaires, the behaviour, I think the behaviour, the appetite, and certainly the ones relating to lower bowel function, carers are very much involved in those.
Excellent. So what about sort of like the quality of life aspects? So, you know, whether there's a reduction in repetitive questioning around food, those sorts of things, will that be captured?
I think that's look, I haven't looked closely at the hyperfat. You may know better than me. It's the standard, it's the same questionnaire that was used in, I think it's the same question. I think the questionnaire we're planning to use is the same one that was used in the liraglutide trial, and it was specially developed for people with Prader-Willi syndrome. And I think it looks at those things. I'm pretty sure it looks at those things. Awesome. But that is very important. We'll make sure it does. So thank you
Awesome.
for raising that question.
Fantastic. Thank you. All right, I think in the interest of time, we'll move on. But so for our audience tonight, if you keep an eye on social media streams, we'll let you know when Tania's trial is up and running, because it would be awesome to have that trial enrolled and recruitment completed as fast as we can, because that's the fastest way to get the answers to the questions. So thank you very much, Tania. Awesome presentation.
Thanks.
I'm going to give you a whistle-stop tour of some of the latest information in Prader-Willi clinical trials. So thank you for joining me for that. To start, we're going to talk about why clinical trials are so important. So clinical trials are the bridge between science and the drugs that are available on the market. The primary role of a clinical trial is to gather data on the safety and the efficacy of a new therapy. And the information gathered from multiple trials is then used during the regulatory approvals process to decide if the evidence base is strong enough to justify using a drug. Basically, is it safe? Does it work? And is the benefit worth the cost? Finally, clinical trials can also be used as an important therapy option, particularly as with Prader-Willi syndrome when there are very few other options. And this is one of the reasons why we work so hard to support and bring clinical trials to Australia. So let's get stuck in. We're going to start with a very brief update on Soleno's new drug, DCCR. So Soleno's most recent trial was a randomized withdrawal study. It builds on the earlier phase 3 trial, which included an open label extension arm. So open label extension means that all study participants had the option of continuing on DCCR once the trial was completed. For a randomized withdrawal study, what happens is that half of the people in the open label extension are randomly assigned to a blinded placebo group, and their medication is withdrawn. So they don't know whether they're still getting the drug or whether the drug's been stopped. What we learned from this was that DCCR is very well tolerated. It's got a great safety profile. The study also showed that the improvements in hyperphagia could be directly attributed to DCCR. And also, importantly, there were improvements in PWS behaviours. So this package of data, Soleno is now going to take that to the FDA. They're going to file a new drug application. They're hoping, last I heard, they were hoping to submit that sometime through the middle of this year, and they anticipate approval in 2025. It doesn't come with any guarantees, and the timeframe will depend on when it's submitted and how long the review process takes, but it's pretty good news. While that's very exciting, it's also worth noting that the open label extension group is a very biased sample. Generally, it will contain an overrepresentation for people for whom DCCR worked. So this waterfall plot, which is the green and blue bars you can see on the slide here, is from the original phase 3 trial. And it shows that while DCCR is very effective for many people, that's these green bars down this end of the graph, it probably won't work for everybody, as indicated by the bars down the other end of the graph. So not so good, very good. And that's why it's so important that research continues and that we have scientists, clinicians like Tania, and companies developing drugs that work on the challenge of hyperphagia from many different directions. So although it doesn't work for everybody, eventually, if we have multiple drugs, hopefully we'll have something for everybody. Now, positive trial data and hopefully approval by the FDA are great. But the really important question is, when will we be able to get DCCR in Australia? So full open access requires the new drug to be approved by the TGA and listed on the PBS to assist with cost. Now, this is time consuming, and it often doesn't start until after a drug is approved elsewhere. So for example, by the FDA. However, the good news is that in Australia, we have very structured processes and pathways for accessing new therapies, including accessing unapproved drugs. The fastest route is via clinical trials. However, clinical trials are often dependent on funding. They can be restricted in location and in the numbers of people who can enrol. There are also schemes that enable clinicians to access unapproved drugs. However, again, there are limits in terms of access and cost. So let's talk about what PWRFA are doing to get access to DCCR. First, we're talking with Soleno and providing them with information about the Australian regulatory system. We're making sure that they keep Australia on their radar, that they know that there is demand for DCCR in Australia, and that we can support them with their application to the TGA for approval and reimbursement. At the same time, we're working with Australian clinicians to identify opportunities to make DCCR available through clinical trials. Once Soleno have submitted their new drug application to the FDA, we will begin working with clinicians to ensure that they have everything they need to access DCCR via the unapproved drug scheme. We see huge potential for the Centre of Expertise to play a critical role in accessing unapproved medications via the authorised prescriber pathway. Finally, the end game is to have DCCR listed on the PBS and readily available for everyone who needs it. To achieve that, we need TGA approval and PBS listing. For this, we'll be calling on you, the Prader-Willi community, for help. As part of the approval process, the Advisory Committee will review the evidence for the safety and efficacy, and they will want to understand the risk-benefit of the medication as well. Your lived experience is critical to understanding the true value of a new drug. We need to help the PBAC understand what it's like to live with hyperphagia and what it would mean to have a treatment. We also need to help the PBAC to understand how to interpret the side effect profile and the risk profile. Life with PWS already involves risk. What are those risks? And how do we balance those risks against the risks associated with taking a new drug like DCCR? What are the side effects? And are they things that you're willing to live with? Once DCCR has reached this stage in the Australian approvals process, we'll be asking for you to share your story. You can play an important role in getting DCCR approved in Australia for everyone. Okay, I'm going to run through the rest of my slides fairly quickly. I'm going to start or move on now to trials that I'm watching closely at the moment and why. So the first two are phase 3 trials. Carbetocin is one that we're familiar with. It's now being, it's part of the Arcadia pipeline of drugs. The reason I'm interested in following this trial is it's the second phase 3, and the data from the first phase 3 trial was really interesting. You may remember this as the agent that was tested by Levo in the CARE PWS trial. The second study is the one that we're particularly excited to be talking about tonight. It's pitolisant from Harmony Biosciences. Pitolisant is used to treat excessive daytime sleepiness and has also shown some benefits for PWS behaviours. It's a phase 3 trial, and it will be primarily looking at efficacy. And this is our big announcement for tonight, is that we heard a couple of days ago that we now have permission to tell you that there will be trial sites for pitolisant in Australia, which is incredibly exciting news. We can't tell you a lot more at this point, but we will be rolling out information as soon as it becomes available. So a brand new trial for Australia, likely to be recruiting in 2025. The remaining 3 trials on this slide are all phase 2 trials. There are a number of other trials happening as well, but these 3 I'm particularly interested in. NNZ-2591 is from Neuron Pharmaceuticals. So Neuron is an Australian company, and they have some very promising results in their mouse models relative to PWS behaviours. What I'm excited about here are 2 things. One is that the data which is coming in from other conditions, Pitt-Hopkins and similar conditions, is showing a really good safety profile. So that's really important, I think, ahead of enrolling our community in the study. And the second reason is that I'm optimistic that we will see some sites down here for the phase 3 trial as well. Nothing confirmed, just hard work on our side. The next trial is also targeting hyperphagia. That's with RGH706 from Gideon Richter. We've worked with Gideon Richter for a couple of years now, and I'm also optimistic that we'll see trial sites down here when they get to phase 3 as well. And the final study that I've listed on my slide here is one that I really, really like because of its scientific mechanism. So it's looking to trigger beta receptors as a way of shutting down appetite and targeting hyperphagia. And what's exciting about that from my point of view is that it's a very different mechanism from the other drugs, which increases the chances that we have something that will work for everybody. So those are the trials that I'm watching. And move to the next slide. Right, so finishing up, I want to take a quick look at the trials that are actively recruiting in Australia now, or in the case of tirzepatide, will be shortly. So the first one is Tania's tirzepatide trial that you've already heard about. The second trial is also being run out of Tania's group and has Dr. Lauren Rice as a co-investigator. This study is a small study, but it's going to be really important. They're looking at acamprosat to see whether that's a possible therapy for PWS behaviours. And as part of the study, the brain imaging is going to help unpack and understand a lot about how people with how the brain of someone with Prader-Willi syndrome responds to these medications. And that has the potential to increase our understanding of what other drugs we might be able to repurpose for use in Prader-Willi syndrome as well. Now, the CBD trial, so CBD is cannabidiol. This trial is being run by Daryl Efron out of RCH. So they have sites in Melbourne and Sydney. This is not a Prader-Willi specific trial. They're looking at aggressive behaviours in anyone with an intellectual disability or any child with an intellectual disability. The reason that I'm promoting this study is that the more people with Prader-Willi syndrome who enrol, the more likely it is that we will be able to generate a pilot data set. And as Tania highlighted, those pilot data sets are the basis for getting larger grants to do the really in-depth studies around safety and efficacy, because clinical trials, unfortunately, are quite expensive. So we hope we will see improvements in aggressive behaviours and other Prader-Willi behaviours. They're also going to do the hyperphagia questionnaire. They've added that on for us. So really important that we get as many people as we can into that trial. The chromosome 15 biobank is really starting to pick up pace now. It's being used to grow and support a number of different research studies, and also underpins David Godler's work, which is around drug discovery. They have sites in Melbourne and in the Hunter in New South Wales that are open, and we're hoping to circle back through Queensland periodically as well. And finally, there are a range of surveys that you can find links for on the website as well, in particular the sleep study or the sleep management survey out of Queensland, as well as the national and the global Prader-Willi registers. Surveys are really important ways of gathering information that we can then use to underpin research and better understand what's going on. So I'd strongly encourage you to learn more about clinical trials and consider whether any of these trials are a good fit for you. As I said before, the faster we can get clinical trials enrolled, the faster we get the answer to the question that the trial's asking. As a bit of an aside, if we can develop a reputation as being able to fill our trials and get good quality data out of them, which is where the right fit comes into it, then we're more likely to attract larger trials down here. So with that, if you would like to know more about clinical trials, you can find listings on the website. Some of the trials have short videos on our YouTube channel, and if there are burning questions, I'm happy to take one. Otherwise, I think we'll respect your evening and pass over. So if you've got any questions, pop them in Q&A or reach out by email later.
All right. Thank you, everybody. I'm so grateful that you could join us tonight, and please feel free to reach out to any of us at any stage, Diane, myself, Debbie or Nicole, whether you're the reason we do this, and we always love to hear from you. Thank you so much. Good night.